Hi, good day, Dr. So. I'm Yan Chi, and in this video, I will propose a GWAS design for the current COVID-19 pandemic. So first of all, what is GWAS? The Genome-Wide Association Study, or GWAS, is a widely used approach in genomic research because it allows for scanning of the human genome as well as analyzing the DNA sequence variations among a vast number of human subjects, whereby only about 500,000 to 1 million out of the 3 billion nucleotides in each genomic DNA will be genotyped and subsequently studied, which in turn identifying the genetic risk factors for common and complex diseases in a population, as well as identifying individuals who are at risk for a particular disease. So how GWAS has helped in the COVID-19 research? From the recently published articles, GWAS has successfully detected 2 genetic loci associated with the severe complication in COVID-19 patients, which is acute respiratory disease syndrome, or ARDS in short. These genetic loci are the 3P2131 locus and the 9Q34-2 locus. However, researchers still lack the knowledge on how exactly does the underlying medical condition like diabetes mellitus associated with a higher risk of ARDS progression during COVID-19 contraction, although there were some assumptions made on the probable etiology for this scenario. Since GWAS had been done previously on identifying the genetic variants for both type 1 and type 2 diabetes mellitus, the objective in this proposal is to design a 2-tier case control GWAS to detect the optimal genetic susceptibility markers for ARDS progression in COVID-19 patients in the United States of America who have a history of either type of diabetes mellitus. There are 7 major steps in this proposed GWAS. First of all, a software program named Quanto will be used to roughly calculate the sample size needed in this study. It is better for the sample size to be large, which is more than 1,000 samples, so as to enhance the power of this GWAS. For the control group, healthy individuals without any underlying health condition and have not contracted with COVID-19 will be recruited. As for the case group, the recruitment criteria would be hospitalized COVID-19 patient with either 5, 6, or 7 scoring according to the WHO establishment, along with a clinical history record of either type of diabetes mellitus. Both groups of subjects will be recruited from the United States of America. In the next step, DNA genomes will be extracted from the whole blood samples of every subject, followed by genotyping of the genomes using Illumina microarray platform, as it has a better specificity as compared to other platforms in the market. The model of the platform in this study is the Infinium Global Screening Array 24 version 3 kit. After all the collected genomes have been genotyped, quality control on both the recruited subjects and genotypes needs will be implemented. The 7 steps of quality control include the identification of missingness of SNPs and individuals, sex discrepancy, minor allele frequency, Hardy-Weinberg equilibrium, heterozygosity, relatedness, and population stratification. Generally, these QC steps serve to check for the sample as well as genotype qualities prior to GWAS analysis in order to minimize the risk of generating false positive or false negative results in the end. In step 4, imputation of genotypes is carried out using a software called Impute2, along with HapMap as the reference panel. This step is essential in statistically estimating the shared information between the genotype samples and the HapMap reference, and then fill in the gaps left ungenotyped in the genome samples. By doing so, the linkage disequilibrium or association between different genetic loci across the autosomal chromosomes can be determined. After that, the P-Link software is used for permutation testing before generating a Manhattan plot. A p-value of usually less than 5 times 10 to the power of negative 8 will be introduced for the significance of the odds ratio of each SNP allele identified, because such a value indicates a significant genotype-phenotype association. Then, the MAGMA software is used to generate a Manhattan plot to visualize the significance of all association signals, whereby the significance threshold is set to be 5 times 10 to the power of negative 10, which is slightly more stringent than the common threshold of 5 times 10 to the power of negative 8. And this is an example on how the Manhattan plot should look like. Replication study will be carried out after the completion of GWAS analysis. This is to ensure that the genetic effects of the identified SNP alleles are equally significant in the GWAS as well as in the replication studies. In order for the results to be valid, both studies need to adapt to the same phenotype criteria for the case and control groups to prevent confusion during result interpretation. Besides, the replication study must also possess large sample size and power to avoid generating false positive results. In the last step, meta-analysis is suggested to compare the result in these GWAS with those generated from previously published GWAS with the same hypothesis being examined in order to produce a more precise prediction on the risk factors for this phenotype. However, as there is absence of previously done GWAS studies emphasizing on the similar hypothesis as in this proposed GWAS design, this step is rather impossible at this stage. As a result, one or two gene clusters across the genomes in the case subjects are expected to be highly associated with the diabetes mellitus related COVID-19 ARDS in this study, with the establishment of a higher than usual significance threshold of 5 × 10⁻⁻10, more claimed significant SNPs could be filtered out in the diabetic subjects, leaving only those accounted for higher significance of association with the phenotype of interest, which is COVID-19 ARDS. With the potential genetic markers in diabetic patients discovered for association with higher risk of COVID-19 ARDS progression, a more accurate underlying etiology for such phenomenon could be revealed, in turn facilitating the decision-making in treatment options as well as the prescription of more effective medications for these patients in combating COVID-19 in future. So that's the end of my video. Thank you very much.